going back to, as I said, to the biology. This is the human genome. It's the code within your DNA that it's in the nucleus of your cells, and it's composed by these 4 chemical letters that we call base, and there are actually 3.2 billion of these letters in each of your DNA molecules. And it is so important because it contains information that it actually that builds what each of us is, and this information is coded in the genes and many other things that I'm not going to talk about here. And actually, since it's this important, scientists started back in 1989 a project that it was called the Human Genome Project, that actually was started by a private consortium and also a public one, with the aim of actually reading the letters of the human DNA in order to get insights or actually understand how the information in the DNA was transformed into human organisms. The project took almost 15 years and cost around $4 billion. But since then, the technology has improved a lot, and around 2005, 2006, few companies developed few new technologies that allowed to, instead of reading the DNA bases, as they did for the Human Genome Project, one by one, these technologies, the main focus of these technologies was fragmenting the DNA molecules in very, very small fragments of around 200 of the letters I mentioned before, and then reading, amplifying these fragments, and then reading the fragments many, many times to get the original sequence. This was done this way because these machines are very fast at reading, but also very error-prone. So in order to be sure that the letter that they read is the letter that was in the original DNA, we have to read it several times. Actually, we call this the coverage. So the higher the coverage, the more times we read each fragment, but also the more data we generate. Then, from this amount of reads, that we call sequence reads actually, we have to reconstruct the original genome of the person, of the individual that we extracted the DNA from. And this is done using actually the reference genome, which is actually the versions of the original human genome that was sequenced, and using this that we call analysis pipeline. So the process works like this. We have, actually, there was some numbers here I'm going to tell you now. So in the beginning we have billions of small reads that on a normal experiment it will go from 100 to 300 or 500 gigabytes for an entire genome of a person. This is why I call this big data files, but not big data, because actually what we usually deal with is big files of a single individual, and the analysis we usually do are on a single individual, but not on many of these. So we usually don't aggregate data of many users. When I said we said, I mean in our company, of many individuals, and then do analysis on it, but we do analysis only on a single big file instead. So when we have all these reads, and then we map them back to the original genome. Once we have all of them ordered, then we look at each position of the original genome, what the machine has read for all of the reads that has mapped there. This schema translates to, so this is how it looks, how it should look, but this is how in reality the bioinformaticians, this is the type of files that the bioinformaticians have to deal with in reality. And the easy pipeline that I've shown before, it's actually, usually it's divided in many steps. Some of these steps are easy to parallelize because, as I said, each read actually is independent. It's a fragment, it's a random fragment of the original DNA sequence. So it's actually independent from each other when we have to map it back to the original reference. So all these steps that actually are related to this mapping process are easy to parallelize, because we can divide all the reads we have maybe in a single file into smaller chunks, and then map them in groups of less chunks. Then the other once this step is completed, we have the next phase of the pipeline, which consists on reading, on deciphering so going back to the original reference and discovering which was the real letter that was in the original genome. So each of these steps actually use different kinds of computations and actually have different computing requirements. As you see in this chart, some of them are very intensive in memory, but actually produce small output files, whereas others are very intensive in this usage, but actually consume very little memory. So, whoops. Yeah. So the thing here is that what this provokes is that at the end, when we have to design this kind of pipelines, we face that at different steps of the pipelines, we need very different resources, and trying to allocate these jobs in a cluster, it might become a bit tricky, depending on the resources we have. So this is what, as I said in the abstract, this is what we have implemented in our company. We have a very small cluster with us now, and then we have defined our workflows in JSON type, kind of JSON files, and then we have a batch wrapper that interacts with a workload manager to distribute these jobs among the computing nodes. Also, all the software that we use, all the scientific software that we use EasyBuild to load the different modules that we need at each point of the pipeline in the nodes, and we load the data, the genomic data from storage within the cluster. But okay, what happens with this? All the workflow design is done in-house and is done for our current cluster. So this means if the cluster changes, if we build more machines, if we change some of the machines, we will have to touch up a lot of lines of code. As I said, different steps have different computing requirements. It's difficult to install, so if we want to change versions of the software that we use at each step, or even if new versions of this software appear and we want to introduce it in our pipeline, this is difficult, and actually most of us, so let's say dealing with configurations of this software and also dependencies that can change actually with different versions it's also painful. So at the end, we need a flexible, a more scalable environment, or we need a way to define these pipelines in a more portable way. And to do so, in order to you to understand which kind of situations we have to deal with in our company, I'm going to very briefly explain the business model. So just for you to understand a bit what we do. So we have users or customers, let's say, for which we sequence their genome, their DNA, and we store it in our system. And then we have these players in the middle, which are pharmaceutical companies, doctors, and also or any healthcare professional, and also researchers that produce not software but pipelines or analysis applications that wants to use this data, to actually read this data, and then extract conclusions that can be useful to provide products and services to our customers. The type of products and services will vary depending on who is the player that is providing them, and also what's the aim of the actual work. So, having said this, what you could expect that happen is that the requirements of these people will vary a lot. So the steps they will want to have in the pipeline can be completely different depending on what they want to do. Or so if they don't vary the steps, what will vary for sure is the versions or the versions or the type of applications that we use. So in order to solve this, many of us, not only us, many other people in this field has been facing this problem as well. There have been efforts in, I would say, two directions. The first one is the workflow definition. So we need a way to standardize the way we describe these analysis tools and workflows, to make these workflows more scalable and also more portable to other environments. So there has been a couple of, let's say, official implementations, which means that they are backed up by some important institutions and so on. And also there has been more in-house implementations of that I will talk a bit more later about Nextflow, which is like a more complete framework on this field. But basically what all these languages do is actually find a way to define each task of these pipelines in a standard way, having what tool we use, what are the parameters that it will run with, which is the input data that they take, and what's the output they produce. And then the second task, so with this we have solved the workflow, the definition. So we have found an easy way for everybody to understand what we want to do. But then the second problem is how we do this. So how we do each task of the workload without facing a lot of trouble. So I'm sure everybody in this room knows what containers do, or what containers are, and what they do, and why are they so useful. So it's easy to see how they can be also useful in our particular case, because with containers we can ship an application, or actually a software of the pipeline, of the workflow, including all the libraries and all the dependencies that this application has. So it doesn't matter if one analyst wants to run this version of this software, and another one wants to run an older version, and they use different Java compilations, or they use different libraries of the system. We will build different containers for each of the applications, and then simply load the container that they need in each time. As I said, so instead of actually using us following the easy build philosophy, we will have a build of each application, and then we will load it. So we will have to, let's say, program a different one for each version, and then load it in the HPC every time we will use it. In this, following the containers philosophy, what we will have is the same, but already encapsulated in what in Docker we call user-defined images. So we will pack all the dependencies of the application with all its dependencies already in a container. This is good because it isolates the HPC platform from the dependency. It helps on the development cycle and the software maintenance cycle, because it allows us to prototype fast and then develop and test the applications easily. It increases, as I said, the flexibility and the portability of the code between the different environments. It allows for a better version control, and actually it helps, and this is very important in the scientific or in this field, to produce reproducible results. So we can make sure that if somebody has obtained some results using a set of instructions or a set of tasks, a set of containers, if they use again the same packets, the same containers, they will get the very same results. And finally, it's also using, because it provides an easy way to share these containers in public repositories. So if most of these tools are used for many people, and probably most of them has been already built within Docker or other providers' containers. So what we will face most of the time is that the tools we're going to build, or we were about to build, that has been done. The containers we were about to build, that has been done by other people already. So yeah, this is an example. So imagine that we have, as I said, one of these third-party providers that comes with a given receipt, with a given workflow. So if we have our pipeline completely Dockerized, the only thing we have to do is just pack each step of the pipeline into containers, selecting the versions they want for each of the steps. It doesn't matter that they use different libraries. They will be already, so these containers will be already prepared to get all the dependencies for the software. And then once we have all the pipeline built into containers, just run it in our HPC facility. And as I said, this is so we have now a way to define what we want to do with the different workflow languages. We have a way to encapsulate each of the steps and their dependencies. Now we need a way to send all these executions to our HPC cluster that will be controlled or managed by Slurm or any of the others, workload manager. And this is what actually our research in the last few weeks, months have been on. So we're looking at different ways to what they basically all together do. They provide as I said, an abstraction layer between the cluster resource manager and the pipeline itself. That together the containers and the list of tasks that we want to do. And okay, yeah. So I've been talking here about the problems we face when we have to do the kind of analysis that we have to do are very similar, but all of them have particular differences that it makes it difficult to automate or to make it interactive. But actually we face another problem, that the amount of workload that we have at different times, it can change a lot. Imagine that during 1 week we have several of these healthcare providers or analysts asking us to analyze stuff of our customers, but then the next week all of them have finished it, all of them have to go to back to the clinics and then analyze the results, and we have no use. So if we have all the analysis and we do all the analysis in our in-house cluster, a lot of times we will face resource limitations or underuse, which means that when we will not be able to allocate all the jobs we want at sometimes, and then in other situations we'll be wasting our computing resources because we will have they will be on but they will not be used. So what's the solution for this? it's to move some of this computation to the cloud, and then pay only for the resources we use. There have been a lot of, this topic is very hot at the moment, and there are a lot of companies building these, the genomics platforms on the cloud to analyze data of anonymous. So they are very popular in research because they allow to analyze anonymized data. So data that belongs to usually patients, because usually there are medical projects, but that no identifier is included within the data that is uploaded. This is good because usually the research institutions can use all this infrastructure only when they need it, and then pay only for the resources that they have used. But they can't do this because all the data, all the metadata, and all the personal data is not included in the data they upload. But what happens, like in our case, we store personal information. We store health information, health reports, and other phenotypical data of all these individuals. And I'm going to give you here two examples of actually the EGA and the DBGAP, which are public genomic repositories that store data for thousands of individuals, but completely anonymized. So the data they store, apart from the genomic files, is completely anonymous, and in theory, none of these patients can be identified by the data that is open at the EGA. And I'm going to compare it with 23andMe, that maybe some of you know, which is a platform that they don't use the same technology as we do, but their model is quite similar. Just for you to see which kind of information each of two initiatives handle. So in the case of the EGA, the usual information we will have for a given individual is information such as the gender, the phenotype, maybe which kind of disease or the individual had, or where the sample was taken from. Whereas for a case like the 23andMe, we will have much more information ranging from the personal details of the birth, where he or she was born, health habits, medical details, and also many other personal information. And as I said, in the case of open data, all this information is available in their websites, but it's always anonymized, and most of the time it's also aggregate. So we don't get specific details of one individual, but instead we get, for instance, ratios or percentages of some trait in a given data set. Whereas personal data, which allows identification of the patients, is collected for each individual specifically, and also is collected on demand depending on the necessities of each study. On one side, for the first approach doesn't allow us to follow these patients after they actually finish their relation with the original research project, whereas in the second case, as we are providing, let's say, a personalized service, this allows us to say, keep an eye on each of the patients from which we have collected information. But this also comes with some problems. As we have all this personal information or all these clinical or health reports, we need to comply with heavy and tough regulations. And actually, what we would like to do with this data is far from what the actual informed consents from the health institutions we deal with allows us to do. And in short, the main problem we have is that the ownership, it's not actually a problem, because at the same time is the focus of our company, is that the ownership of the data So the data belongs to the patient. We cannot do anything that the patient doesn't allow us to do. So, but on the other side, the patient can do whatever he or she wants with the data. So what this becomes at the end is that every time we need to access, or every time we need to read, write, or analyze any related patient data, we need to ask permission to the patient. And this also give us some other constraints, like for instance, clinical data cannot be stored in a public cloud. So this would be a very convenient and cheap way to adapt to the changing demands of our resources, but we have to keep them in a, let's say, controlled and secured location. As I said, any data cannot be handed over, cannot be read without the acceptance of the informed consent. So this is the solution we have come out to solve this limitation. We have a private data center. So it's in a locked door. Where, like physically, we are the only ones allowed to access, or our systems administrators are the only ones allowed to access that building where the data center lies, where we stored the personal data and all the clinical information of our users together with the genomic data. And then we have an instance in a cloud, in a public cloud actually, where we store the identified genomic data for the users. So what we call disassociated raw data. Then we do all the analysis we need in our private instance, and then when we need to run anything that let's say outpace our capabilities at the moment, we move this computation to the cloud. Yeah. So as I said, the clinical data and personal identifiers are stored in our private cloud, or in our private data centers, and raw and processed genomic data is stored in both sides, here and here. But since the raw data, the raw and processed data stored here can be linked to the personal data that is stored in the same system, but not for the public instance. And basically, this is how the data flows within the system. So when we receive the raw data, we do the mapping I mentioned at the beginning, and once we have these aligned reads, we save two copies. One without any identifier or personal data in the cloud, and one in our private archive, which is actually linked to all the databases that contain information for the patients. Apart from that, everything is encrypted when it stays on the disk, and it's only unencrypted when we are running processes, where we are running analysis on this data. Moreover, when the analyst or any professional that is authorized to access the data of the clients wants to access the data, they use, we call it a host-proof methodology, which consists in, we also encrypt the data, so they are the only ones allowed to actually read those results. And this leads to a schema like this, where we have the delimiter zone with all the external, the web app and the API they use to interact with us. And then we have two instances of computing nodes and genomic data storage, one in our private cloud and another one that we can turn on and off depending on the demand. Yeah, so what I've been explaining this, it's called actually cloud bursting, and it consists on this. We can adapt on the, so we have our Slurm workload manager that can interact with the API. it talks with the API of the cloud provider. It's missing a column here. It talks with the API of the cloud provider and just turns on new computing nodes using the images we have in our repository. And when these demands go down and it detects many of the computing nodes in the cloud are idle, it just turns off these instances that it just opened, and then we go back to the initial situation. And this is actually a summary of what I explained. As you can see, this is the infrastructure in our private environment, and then it's connected through a VPN to the cloud. At the moment, we work with SoftLayer, which is a company that belongs to IBM, but we can more or less easily migrate this infrastructure to any other cloud provider. All the connections that our staff does through either to one or the other environment is done through the VPN as well, and we use several methods to ensure that we are accessing the infrastructure but not anybody else. And that's all.
Thank you, Marc.